Okay, welcome to Chapter 5. We're going to be covering DNA, gene expression, and biotechnology in this section. This really is the heart of all of life, really is the genetic makeup and background of each of us. And we're going to discuss really how the code is arranged and how it's actually utilized to create who we are. So let's start to dig into this material, and I hope you enjoy it. We've already covered a lot of this in class, but I'm going to reinforce and help you build a good basis. So what we're going to talk about, of course, is describing what DNA is and what it does, explain the process of gene expression and the collaboration of nature and nurture. What do we get from our genes, and what do we adapt to our environment, that sort of thing. Explain the causes and effects of damage to the genetic code. Describe biotechnology and its implications for human health. Discuss, of course, the biotechnology in agriculture and what agriculture and biology may do. Of course, feeding the 7.3 billion people we have on the planet. Discuss biotechnology today and tomorrow, so we're going to discuss aspects of these things. So let's start off. What is it and what does it do? So knowledge about DNA, of course, actually can be applied for even the legal justice system in itself. And it's actually done more good for releasing folks that have been improperly arrested in terms of evidence that maybe not conclusive, but was considered for a state's evidence. It was conclusive for a jury, but did not have a complete picture. But now with DNA technologies, it actually tells who might have been or who might not have been at a crime scene. So in this case, this Julius Ruffin was 27. He worked at Eastern Virginia Medical School. Got into an elevator, lost more than 20 years of his life. Several weeks earlier, a nursing student at school had been raped by an attacker who broke into her apartment. When Ruffin got to the elevator, the student thought she recognized him as her attacker had called police. And they, of course, based on that testimony, I mean, it is one person's statement over another. So when she testified, of course, she was convinced, an eyewitness, but he maintained his innocence to no avail until 2003. The State Division of Forensic Science performed DNA analysis on the swab of evidence that remained from the investigation. The analysis from perhaps a rape kit or some aspect of that proved that he was not the attacker. So he was vindicated from that crime. Without that technology, without that evidence, he'd probably still be serving. So that's a very good and positive effect of using a technology like this. So what is the most common reason why DNA analysis overturned the incorrect criminal investigations? And you want to speculate on that? Well, his case is not really out of the ordinary. Many people have been freed from prison, and it has been due to the DNA analysis that overturned a lot of these cases. So about 80% have been convicted of sexual assault. 28% were convicted of murder. In more than 3/4 of the cases, inaccurate eyewitness testimony played an important role in the guilty verdict. So we're gonna take a closer look now into DNA. Selfish dictators may owe their behavior partly to their genes, according to a study that claims they found a genetic link to ruthlessness. So that's the rude gene. Whether a man has one type of gene versus another could help decide whether he's a good husband material. Well, that may be true. I'm not really sure. The importance of DNA goes far beyond its function. as an individual identifier. However, the information carried within the molecule, which is organized in individual units called genes, is among the most important of all the biological knowledge, and is witnessed by these headlines that often are in the news. So a lot of times, though, we have to take in consideration that there are studies done with identical twins, and even though they had identical genetics, a lot of times they grew up to be quite different individuals. So although we can contribute some of the aspects of this to genetics, some of it is the influence of the environment or experiences that we have during life. So the age of the gene, a lot of top covers reflect human development various times in stages, and we see the Time magazine, what makes us good, what makes us evil, DNA is not your destiny, and the Discover. So these are all accounts or topics that we think about. So the age of the gene. So what is this entity that appears to exert influence over everything in our grooming behavior to our propensity for suicide? We explore these issues and more in this chapter course, beginning with the structure of DNA itself, how it contains information for producing organisms, and then we'll talk about how modern manipulations of DNA really have a huge implication for our future, our health, and agriculture. And so DNA is a molecule that all living organisms carry in every cell in their body. Unique in virtually every person, DNA can serve as an individual identifier left behind as we go about our lives. This is a fact that is used increasingly to ensure greater justice in society, and as we talked about with convicted crimes. The DNA molecule contains instructions for the development and functioning of all living organisms. There's two important aspects or features of DNA. DNA contains the instructions on how to create a body and control its growth and development. So not only does it encode the genes for the expression of the, let's call it the firmware, or the all the pieces and parts that make us up, but also it regulates, it turns on and turns off during the different stages of development, and these are very important aspects. The instructions encoded in the DNA molecule are passed down from our parents to their children. So as we know this, but keep in mind that there's 3 new mutations that occur every time we do pass our genes from the parents to the offspring. Some of them may be apparent, some of them may not be. So in the beginning, in the 1900s and continuing through early 1950s, a series of DNA experiments were conducted that showed 2 important features. First, it contained instructions on how to create a body and control its growth. The second, the instructions encoded in the DNA are passed down from the parent to the offspring. Here are some diagram of drawings that Watson and Crick had proposed. The big advantage that they had, of course, is that they moved in location. If you recall, we had a video during class that was an interview with Doc Watson. If you did not catch that for any reason, the link for that is in the folder for Chapter 5, and I would strongly suggest that you view it, because it really gives you straight from the author's mouth. Although he's up there in the years and a little bit cagey, I think you'll enjoy that video. So if you didn't get a chance to see that, it gives you an insight and a little bit more of an understanding from the genius that worked this out. So the double helix, what we have now learned about the structure in the 1940s, this was a huge development, because prior to this, no one really knew what was going on with the development of living things, and even Leonardo da Vinci thought sperm cells contained little dinky or very small people inside the sperm head, and that person would then incubate in the mother's womb, and voila, you have a child. Well, he was right to a very small amount, I guess you could think about. It had all of the genetic components, but it wasn't a small little human. It was half of the genetic component, as we'll learn later on. So a nucleotide is a unit of DNA molecule that contains very important three components: a phosphate group, a sugar, and a nitrogen or nitrogenous base, or a nitrogen-containing base. And for an example on the screen here, we have adenine. And if you look, the phosphate group has the double-bonded oxygen with three oxygen groups, one of which is bound to the next carbon, of course, that make up the sugar, and then the sugar is bound to the adenine. So how does this arrange in DNA? If you notice, the phosphate groups are the backbone, so you can see the yellow portions over here on the sides. They go up and down the sides of the backbone. And then, of course, there's the sugar, and then there's the base. The bases are what align and hold two strands. This is one of the key discoveries that Watson and Crick made, was that DNA is a two backbone component of DNA. Linus Pauling thought it was three, and others did as well. And then found that it was only two strands of backbone. So, and they are held together by these nitrogenous bases. And we see that adenine binds with thymine, and cytosine binds with guanine. So the G's and C's bind together, and the T's and A's. Now we could reverse that, and we could have the thymine binding to an adenine, but it's the same. Adenine and thymine binding is just held by the different strands. So we only have four different possibilities, and you think about that. All of life is really comes down to four bases. So a double helix, for review, consists of individual units called nucleotides, which have three components: a molecule of sugar, a phosphate group, and a nitrogenous base. If you look at the figure in 5.4, you can see that it's a spiral staircase, is one way of thinking of it. Each of the bases are binding to each of its a compatible base that it would bind with, so T's and A's and G's and C's. And sugar is always deoxyribose, and the phosphate molecule is always the same too. It's the shape of the backbone molecule that causes DNA, the ladder, to twist. So the sugar, phosphates, and bases, A, T, C, and G, are the base pairs. So these alternating sugars and phosphates hold everything in place, but they play only a supporting role. The rungs of the ladder are where things get interesting. Attached to each sugar, protruding half of the rung of the ladder, is a molecule called a base. Each sugar always has a base attached to it. When discussing DNA, these bases are usually referred to their first letters A, T, C, and G: adenine, thymine, cytosine, and guanine. The other side of the ladder also has bases protruding from the sugar. The base from one side of the ladder binds via hydrogen bonds, or the van der Waals forces, to the base from the other side. Together, these base pairs form the rungs of the ladder. They just don't pair up at random, though. Every time C protrudes from one side, and it binds to G on the other, and G always binds to a C, and vice versa. Every time a T protrudes from one side, it binds to an A on the other, and of course vice versa. For this reason, the DNA molecule always has the same number of G's and C's and the same numbers of A's and T's. Because of these base pairing rules, it's also true that if we know the base sequence from one of the strands of the DNA molecule, then we'll know the complement strand of the other, because the A's always bind with T's, so we can determine the two strands. So the DNA sequence is described as a sequence of bases on only one of the strands. DNA is a nucleic acid. It's a macromolecule that stores information. It consists of individual units called nucleotides, which a nucleotide is composed of a sugar, a phosphate group, and a nitrogenous base. The DNA structure resembles a twisted ladder, with a sugar and phosphate group serving as the backbone of the molecule, base pairs serving as the rungs. So genes are sections of that DNA that contain instructions for making proteins. So why is DNA considered the universal code for all of life? Well, everything that we know of that's living is consisted of DNA. So we find this true on all organisms. So one of the most amazing feature is this fact that the building instructions, or the cells and structures for virtually every single living organism on Earth, is encoded by DNA. Furthermore, of course, all these living organisms use the same molecules to transmit. So DNA in any organism utilizes the same universal language. So A, T, C, and G are universal. You can think of the organism's DNA like a cookbook. Just as a cookbook contains detailed instructions on how to make a variety of foods, such as French toast, macaroni and cheese, chocolate chip cookies, an organism's DNA carries the detailed instructions on how to build an organism and keep it running. And just as a book can be viewed as a sequence of letters, and the book's meaning determine which letters are strung together, a molecule of DNA can be viewed as a sequence of bases. In a book, letters don't have much meaning on their own. When they are put together into words and sentences, their order holds a great deal of information. Similarly, the sequence in which bases appear in a molecule of an organism, the DNA makes up a secret code that holds the detailed instructions for the building of the organism. Either it's a one-celled amoeba to a giant oak tree or to a human being. And so we can see in this diagram, as we drill down to the sequences, we go from the genome, which is the complete set within the DNA, or of the chromosome, held within the nucleus of a eukaryotic cell. Chromosome, one of the unique pieces of the DNA, which is circular in prokaryotes, but is linear. In our case, we have 23 pairs that together make up an organism's genome. Chromosomes vary length and consist of hundreds of millions of base pairs. Humans have 23 unique chromosomes and have 2 copies of each, one from our mother and one from our father, for a total of 46 chromosomes. 23 pair, 46 chromosomes individually. A gene is a specific sequence of DNA, on average about 3,000 bases long, and contain information necessary to produce every part of the protein. The full set of DNA present in the individual, again, is called a genome. In prokaryotes, including bacteria, this information is contained within a circular piece of DNA. In eukaryotes, including humans, this information is laid out into these long linear strands of DNA, and we call these chromosomes. In humans, I have about 3 billion base pairs divided into 23 unique pieces of DNA, or chromosomes, one from our mother and one from our father, for a total of 46 chromosomes. So we have the pairs of 23 and 46 chromosomes altogether. So try to keep that distinguished there. And 6 billion base pairs in every cell. That's a lot. The number of chromosomes vary from species to species. Corn has 10 unique chromosomes. Fruit flies have only 4. Dogs and chickens have 39 different chromosomes. And goldfish have 47 chromosomes. Individuals of each of these species inherit one copy of chromosome from each of its parents, just like it does in humans. Genes, a sequence of bases in a DNA molecule that carry the information necessary for producing a functional product, usually a protein molecule or RNA. So alleles within a species, individuals sometimes have alternate versions of a gene for a given protein. These instructions can result in different versions of the same trait. Allele is an alternative version of a gene that codes for the same trait. So we have allele 1, allele 2, and allele 3. And in this case, each, when the protein is produced, have a slightly different color of the flowering, the flower itself. So you can see that the different alleles encoding for flower color, of course, are different. So each gene in the instruction set for producing one particular molecule, usually a protein. For example, there's a gene that encodes for fibroin, the chief component of silk. this is different gene that encodes for the triacylglyceride lipase, an enzyme that breaks down a dietary fat. So that triacylglyceride lipase is that enzyme that breaks down a dietary fat that we have to have. When a species, an individual sometimes has a slightly different instruction given for that protein, and the instructions can result in a different version of the same trait. These alternate versions are called alleles, as we just discussed, and the single feature of an organism is referred to as a trait. So a simple hypothetical example would clarify the meaning, the color of the daisy's petals, as a trait. So as we already discussed, the instructions for producing this trait are found in a gene that controls petal color. So one allele may specify the trait of the red, the other might be white, and then yet another yellow. So we have one allele for eye color for fruit flies and carry the instructions for producing a red eye, while another one slightly is different, which we call as a different allele, may be instruction for brown eye. So we have three different examples here of the fruit flies and the different color petals. So we have the trait, and then different versions or different flavors of that trait are called alleles of that gene. So we'll come back to that. The different traits that we talk about are different genes, but when we talk about the same trait, that same gene that might have alterations like the color, again, would be alleles. I just want to make sure we have that. DNA is a universal language that provides the instructions for building of all the structures of living organisms. The full set of DNA an organism carries is called its genome. In prokaryotes, the DNA occurs in circular pieces. In eukaryotes, the genome is divided among smaller linear strands of DNA called chromosomes. A gene is the sequence of bases in DNA molecule that carries the information necessary for producing a functional product, usually a protein molecule or RNA. And not all DNA contains the instructions for making proteins. We have human, there's 3,400 various sizes there. The newt has 84,000 genes. Fruit flies 180. Onion has 18,000, and amoeba has 670,000. So estimated genome size, millions in base pairs. So 3,400 actually is 3.4 billion base pairs in the humans. So that would be 84 billion in the newt. Fruit fly has 180 million. So the onion has 18 billion, and then the amoeba has 670 billion. That's a lot, but anyhow, we'll continue on. Not all DNA contains the instructions for making proteins. It's debatable whether humans are the most complex species on the planet, but surely they're more complex than an onion, even though the onion has all of those base pairs. But we're not, if you measure the complexity by the amount of DNA an organism has, an onion has more than 5 times as much as a human. So we don't fare any better compared to other seemingly simple organisms either. A salamander species has about 25 times as much DNA. So that sure makes things very interesting. So doesn't that make the onion more complex? Well, comparing the amount of DNA present in various species is really a paradox. There does not appear to be any relationship between the size of the organism's genome and the complexity. Among species of seemingly similar metabolic and behavioral complexity, there can be huge differences in the genome size. Among fish, for example, there's more than 3-fold differences among species of the surgeon. So what's going on? The proportion of DNA that encodes for genes in humans is 2%, and the fruit fly is 19%, roundworm is 25%, and E. coli is 90%. So the percentage of DNA that codes for proteins increases amongst different species. Coding versus non-coding DNA. We have gene 1 and gene 2, but we have interspersion there, regions of DNA that do not code for protein. So 75% of non-coding DNA is found between genes, while 25% of non-coding gene DNA is found within genes. Most DNA in eukaryotes does not code for any proteins. So that makes things quite interesting. So this brings up this concept of introns, or intervening sequences. Non-coding regions of DNA may take up the form of short or long sequences that are repeated thousands of times. May also consist of gene fragments, duplicated versions of genes, and pseudogenes. So just to recap, an intron is a non-coding region of DNA. Sometimes it takes the form of short sequences that are repeated thousands or even hundreds of thousands of times. Other times, the introns are slightly longer repeated sequences. Occasionally, the non-coding DNA consists of gene fragments, duplicate versions of genes, and pseudogenes, which are sequences very similar to the actual genes, but with a few slight alterations that make them lose their protein coding ability. So they're dysfunctional genes. At the end, the presence of this non-coding DNA remains a big mystery. We don't really know. All the extra DNA may serve some purpose. Perhaps it's a reservoir of potentially useful sequence, or it may have some function in regulating when genes are turned off. But we're not certain what the non-coding DNA regions do. And a lot of folks label it junk DNA, but I wouldn't rush to that one. I don't believe nature puts in things that are not used. We find this in many cases. So I would use the junk DNA very sparingly, because I think it says the jury's still out, and it's too early to assume it has no function. Well, we're finding functions all the time. Only a small fraction of the DNA in eukaryote species code for genes. The function of the rest is still a mystery. So how do genes work? We have some terms to get under our belt. Genotype is all the genes contained in an organism. The phenotype is the physical manifestations of what we see as a result of these instructions. So genes in the strand of DNA are a storehouse of information as an instruction book, but they're only one part of the process. All of the genes in the organism has its genotype, like the recipes in a cookbook. The physical manifestations of the instructions, the organism's phenotype, are the cookies, the macaroni and cheese, the French toast, and all the other foods described by the recipes. And just as you've assembled ingredients, mixed them, and then baked the dough to get the cookie, there are several steps in the production of the molecule, tissues, and even behaviors that make up. So how do genes work? As an overview, we have really two sections that we need to talk about: transcription and translation. Transcription, the sequence of a gene, or for a gene, is copied from DNA to a middleman molecule called the messenger RNA, or mRNA. So inside a nucleus, we take the cookbook and read from the cookbook, and we copy onto a recipe index card things or the information contained in the original region of that cookbook. Then we utilize that process, by the way, is we're transcribing it to an index card, if you want to remember it that way. Then we take that index card, or that messenger RNA, and we translate it into baking ingredients, for an example, to make the cookies. So we translate the sequence for a gene by the recipes, and actually act upon that and build whatever it is that that messenger RNA or that recipe calls for. And the recipe calls for combining these components to make cookies, where these components are built to make a particular protein. So instead of the eggs and butter and milk, we have the various amino acids. We have 20 of them that can be assembled to make various different sizes of proteins. So how does a gene or sequence of bases within a section of DNA affect the flower's color, or the shape of a nose, or the texture of a dog's fur? The process occurs in two main steps. We have the transcription, which is the copy of the gene's base sequence, and then translation, which is that copy is used to direct the production of a protein. So if you go back and look at figure 5.11, this presents the overview of the processes of transcription and translation. So you keep in mind, in transcription, which occurs in the nucleus in eukaryotes, the gene's base or sequence or code is copied The DNA is not directly used. It's sort of like the magic or the most important volume of data, and we don't want to disturb that in any way, so we only make transcriptions from that or transcribe that into a recipe. And that middleman molecule is called a messenger RNA, and I think it's a good use of middleman to remember the M for messenger RNA. This is like copying the information for a chocolate chip cookie recipe out of the cookbook into a piece of paper. In translation, messenger RNA moves out of the nucleus into the cytoplasm of the cell, where the messages are encoded for the messenger RNA molecules used to build proteins. So the genes and the strands of DNA are a storehouse of information and instruction book. The process by which this information is used to build the organisms occur in two main steps: transcription and translation. Transcription, in which a copy of the gene's base sequences is made, and translation, in which the copy is used to direct the production of a protein from various amino acids. So we've now covered at least the initial phase of DNA technology in itself, and we'll move on now to other sections of the DNA technology as we'll describe. So we'll be back.